Hi, my name is Maria Marco. I'm a professor in the Department of Food Science and Technology at the University of California, Davis, and I'm also on the ISAPP Board of Directors. And in this talk, I'll be describing to you the goals and outcomes of a consensus panel led by ISAPP this past year. So what are ISAPP organized consensus panels? These are panels composed of academic experts from multiple disciplines. Consensus statements are the outcome of consensus panels, and these are reports that are intended to be authoritative, independent of industry influence, and published in high-impact journals in order to reach a wide audience. The goal of consensus panels is really to provide some guidelines for stakeholders on important ISAPP-related subjects. And our stakeholders include consumers, researchers, clinicians, and other healthcare professionals, industry, and regulators. There have been a number of consensus panels, including others besides the fermented foods consensus panel you'll be hearing about at the ISAPP meeting this year. In the past, we have two published reports. One on probiotics was the first ISAPP consensus panel report, published in Nature Reviews Gastroenterology Hepatology in 2014. And then the second one to follow was one on prebiotics, published a few years later. Both of these papers have received a lot of attention, are highly downloaded and cited. So we're excited to be going forward with the fermented foods consensus report. Here's the panel that assembled in London in September last year. We had representatives from many parts of the world, including Asia, Canada, the US, and Europe. I particularly want to point out Bob Hutkins, whose name is here in bold on the slide. Bob chaired this consensus panel with me, and the two of us worked closely together to develop the program and the manuscript that followed. I also want to point out Katrina Ray, who is the chief editor at Nature Reviews Gastroenterology and Hepatology, who was also in our room and contributed to the discussion as well. So why a consensus panel on fermented foods? We all know of fermented foods. We know that they have been made for thousands of years, and we also might know that they are increasingly popular. And so in the past 20 years. However, we also noticed that there are numerous misunderstandings, misnomers, and questions about fermented foods, even among scientists. For example, you might have heard that if live microbes are absent in a food, that food couldn't have been fermented. Other misunderstandings include that fermented food-associated microbes are all probiotic, that these microbes colonize the digestive tract, and broadly, that fermented foods improve human health, that being all fermented foods. So we wanted to take a look at this, these different questions, and really recenter on the topic of fermented foods in our consensus panel and statement. So we reached the following topics to cover in our panel discussion. These topics came out of pre-meeting emails and phone calls, and we developed a very detailed agenda prior to meeting in London so that we could really focus in on these points during our panel. So we wanted to look at the definition of fermented foods. We wanted to review progress on what we know about the microbes and processes required for making fermented foods. Take a look at what we know about safety and risks of fermented foods. Evaluate the strength of evidence supporting health benefits of fermented foods, including epidemiological, clinical, and mechanistic evidence. And also review regulatory aspects and the status of fermented foods in national dietary guidelines. So for the definition, we return to this question of what is a fermented food. If you open a textbook or go online, you would find probably many definitions of fermented foods. And as a panel, we thought it was very important to provide a clear definition that we all agreed to in our report. So the definition we reached is the following. Fermented foods are foods and beverages made through desired microbial growth and enzymatic conversions of food components. So I've highlighted a couple parts of this definition that I want to take you through. First of all, for simplicity, when I refer to fermented foods, I also mean beverages. And that's just for simplicity in this discussion. The other important aspect here is the desired part. And we had a very lively debate around this definition as a whole, in particular this word, where we discussed numerous other types of words we can include in here: intentional, desirable. We voted in the end and reached desired. So we're talking about desired microbial growth and enzymatic conversions. And we're satisfied with this definition because it encompasses all variety of fermented foods and beverages. We're able to distinguish fermented foods from microbial food spoilage, or the outcome of microbial growth that's undesirable, undesired, not wanted, and therefore the food wouldn't be eaten. And it also allows us to clarify what is and what is not a fermented food. Again, to help clarify to the public and to other stakeholders the differences. For example, adding vinegar or pickling fruit or vegetable would not be a fermented food product. Or using chemicals to leaven bread. We would have a food with a fermented ingredient in low quantities, so that food would not be a fermented food. Or a food where a live microbe is added to the final product, and that microbe isn't responsible for the properties of those foods. That would also not be considered fermented. In our discussion, we talked about advances in understanding of the microorganisms involved in making fermented foods. There is a very rich history in the microbiology of fermented foods going back to Pasteur, but we wanted to highlight what we know and then where we're going with the science. So first of all, fermented foods use a variety of organisms: lactic acid bacteria, acetic acid bacteria, bacilli, other bacteria, yeast, or filamentous fungi. Of course, the exact desired composition depends on the food being made. And we can imagine, and we know also that there's a variety of microorganisms down to the strain level. So between different food fermentations, we want to acknowledge that. We noted that industrial starter and adjunct cultures are available for some, but actually not many fermented foods, indicating we have opportunity for growth in developing these starter cultures for controlling fermented foods in a better way. We looked at the methods we're using to characterize the microorganisms in fermented foods, and we acknowledged that culture-based methods remain the gold standard for characterization of microorganisms in fermented foods to the strain level. However, we appreciate that holistic omic methods are really shedding a bunch of new information on the microbial community dynamics in fermentations, using approaches like genomics and then metagenomics, metatranscriptomics, and so on. Another important facet in thinking about fermented food are the processes involved in making that food. We know that the microorganisms are, of course, critical, but there's also the ingredients in those foods, the time dependency of the ferment, and the physiochemical parameters like temperature, pH, water activity, and so on. But beyond this, we appreciated that increasingly principles of ecology and evolution are coming into the study of fermented foods to help provide a richer understanding of the processes that are involved. Principles including dispersal, drift, selection are all involved in making a fermented food. And we're able to do much more once we can reconstruct the microbial community of a fermented food and then relate it back to the wild or the natural ferment. So we're able to ask questions of which microorganisms are available to build a food microbiome, and that incorporates elements of dispersal. How do the microorganisms fit together through their interactions? And how do microbiomes in fermented foods change over time? And we can apply evolutionary principles, for example, to understand domestication processes that have led to the industrial starter cultures that we have today. We discussed safety and regulation surrounding fermented foods, and we acknowledged that when properly made, fermented foods and the bacteria and fungi responsible for their manufacture have a long history of safe use. We do acknowledge, though, that there are some potential risks, particularly for low acid fermented foods where foodborne pathogens are able to survive. There is the possibility for antibiotic resistance genes amongst the fermented food microbiota, and that some fermented food microorganisms can make biogenic amines and mycotoxins, underlying the need to have a test for these genetic and biosynthetic capacities to make sure our starter cultures do not have those. So understandably, regulations on fermented foods are mainly concerned with food safety. We do see opportunity for new regulations to develop new standards of identity for fermented foods. And we're going to see that's possible as we better understand the core microbiota needed to make a fermented food. And so that we came back to when we look at the microbiome or the metaomic approaches that are being used to understand fermented food, the microorganisms involved. So next we also in our group, we talked about the differences between fermented foods, and we could categorize them in various ways, but we want to highlight here whether live microbes are present or absent, going back to the confusion there is amongst stakeholders of what is and what is not a fermented food. So for example, where we eat a fermented food, we expect live microbes to be present, would be yogurt. But there's a whole bunch of fermented foods we would not expect to have live microbes. For example, in bread, in coffee that's fermented and then roasted, and chocolate in the same way, or foods that are pasteurized or filtered, for example, soy sauce. For those foods where we would expect to have live microbes, that's where we expect, we find that there is some confusion over between the differences between probiotics and fermented foods. So for probiotics, these are live microorganisms that when administered in adequate amounts confer a health benefit on the host. And if you look at the bottom of the slide where there's a green check in the red box, that's to show what we expect a probiotic to have. We expect a probiotic food to have the probiotic be alive at the time of consumption. The probiotic should be at levels demonstrated to confer a health benefit, of course, to be safe for intended use. And we expect that the probiotic would be characterized down to the genome level. Fermented foods, on the other hand, are not that. They don't have to be that. So you can see in the blue boxes on the bottom of the slide where there's an X, we don't necessarily expect that. We of course expect that the microorganisms used to make the fermented food are safe. But there is not the expectation that they have a health benefit. So in this discussion, we talked about what would be appropriate labeling or claim that could be made on a fermented food. So there's options. One, if a living microbe or microbes are not present in the final product, we could say these are foods made by fermentation. If the food contains live microbes, but for which we have no evidence that those microorganisms have a role in human health, we could say these foods contain live and active cultures. So those are pretty clear, simple messages to get across. Where it gets a bit more confusing and challenging is when you have this green box here in the middle called probiotic fermented foods. So for a probiotic fermented food, there is the possibility where you could check all of the boxes that we would check for a probiotic, but we wanted to characterize this a bit further. So in this slide, we talk about the two possibilities for a probiotic fermented food. First, if there's no strain-specific evidence, there is a possibility for some fermented foods containing live microbes to have a claim contain probiotics. This would only be appropriate for foods containing known strains, again, defined down to the genome level, from well-studied species that confer probiotic health benefits via principles of core benefits. And these core benefits are discussed in the ISAPP consensus paper on probiotics. The deepest level of evidence and what you could say in a claim would have if we would have strain-specific evidence. There we could say that would be a probiotic food. And with that, it would come with a claim. For example, helps to reinforce the body's natural defenses. However, claims like that should only be made if they're supported by evidence for the bacterial or the microbial strain present. We next moved on to discuss dietary guidelines and health benefits of around fermented foods. And we noted, although fermented foods are generally regarded to be healthy, they typically are absent from dietary guidelines. And we had looked at dietary guidelines from around the world. There are some exceptions on certain foods. For example, dairy gets particular attention. We also noted that except for yogurt and cultured dairy products, which have over 20 RCTs, there are very few clinical studies performed on fermented foods. Through the literature, it should be noted that there's very few RCTs, for example, for milk kefir, kimchi, sauerkraut, natto, vinegar, sourdough bread. There are RCTs, but more are needed. For some fermented foods, we have none, for example, kombucha. Therefore, we point out that a path to establish health benefits of fermented foods using population-based diet and health databases and new RCTs. And for that, we could look at categoricals of fermented foods, for example, yogurts or sourdough breads, or we could look at specific products. Importantly, if we're to also understand the benefit of these foods to our health, we'd want to look at the mechanistic basis for their health benefits. And so we outline various ways that this could occur. For example, through fermentation, we could be removing toxic or unwanted compounds. For example, reduction of lactose in dairy fermentation. Through fermentation, we might also get the synthesis of new compounds that would be active in the digestive tract, for example, vitamins. When these fermented foods contain live microbes, we might expect that they interact and alter the gut microbiota. And even the fermented food itself, beyond the microorganisms, or for example, if the food is pasteurized, we might also have an effect on the gut microbiota. Through the gut microbiota, or perhaps just directly from the constituents of the fermented food, we could have also impacts on the immune system and broadly on systemic effects throughout the body on other organs. So in conclusion, fermented foods, we know, have been staples of the human diet for centuries, and even today, they count for approximately 1/3 of the human diet. We continue to see advancements in fermented food science, particularly as we find more evidence of the core microbiota through fermentation using omic methods. So we expect these types of studies to continue and to provide more depth and guidance in the future for controlling for fermented food processes, and possibly even coming up with biomarkers we could use to assess the potential of these foods to benefit human health. We find the need for more human and mechanistic studies designed to address health benefits of fermented foods. Studies will be useful for establishing their roles in promoting human health and importance in dietary guidelines. So I'm pleased to report the panel's findings were compiled into a manuscript. This consensus report manuscript is now under review at Nature Reviews Gastroenterology and Hepatology. Again, I want to acknowledge my co-chair, Bob Hudkins, who was instrumental in pulling this panel together. Also Mary Ellen Sanders, who was on our panel, and her guidance in ISAPP also has been critical to the development of the panel and the manuscript. I also want to acknowledge all of the great panelists we had. Their expertise really helped to bring it all together. And with that, I want to thank you and talk to you in the future.